All right. So when we in the session from today morning, we spent a lot of time talking about how ideas or information theoretic concepts show up in a non-equilibrium self-assembly, and we also sort of fleshed out or sketched out this idea of proofreading. In fact, we used proofreading as sort of a motivational example to get into self-assembly and to think about how concepts like relative entropy, which we had discussed purely from information theoretic standpoints, actually show up in if you happen to do a thermodynamic accounting of all the fluxes that go into a non-equilibrium self-assembly process, and this we sort of realized that this is not just a theoretical result. Well, it is, but it also has a utility. So the bounds that come out of these considerations can actually predict the kinds of structures and compositions that are formed, and so you can hope to do make meaningful predictions from all these things. And so where does this leave us? It sort of gives us this idea that when applied, if you were to apply these concepts to biophysical systems, you can come to the conclusion that biology does indeed care about bits. It sort of cares about these information theoretic concepts. These are not just abstract tools, but rather they can actually quantify how information is transferred or processed in noisy systems. I do want to sort of point out that the models I had were don't come close to the complexity of a real biophysical system, but these were just toy models that sort of illustrate these ideas. But these toy models do have non-trivial correlations, and so it's one can be hopeful that one can take these kind of ideas and apply them to real life systems and see what happens. So what I'll do in the second part now is I'll talk about information processing. So in the lecture today morning, I sketched out the kinetic proofreading model, and I'll put it up again just to refresh your memory, because it is after lunch and it's a long day. So I'll put up the slide again, but I'll talk mainly about information processing. I'll talk about the proofreading model. I'll talk about how things like dissipation or energy consumption show up in these proofreading models, and I'll try to sort of give a feel for or sort of convey some ideas related to what kinds of steady states show up in these biophysical models, what kind of properties they have, and how the whole thing comes together, how systems can use non-equilibrium forces to sculpt landscapes and achieve desired function. Okay, so that's sort of where I want to go. Again, I want to, in these three lectures, obviously I can't do justice to this vast new area which talks about biophysical information processing and control. I have some suggested reading that's been put up. So this is a set of 3 papers which I think are, they're very illustrative, they're easy to read, they're fun to work through, and I hope that sort of gives you a flavor for how people are thinking about these problems in various different fields. Okay. So again, I put this up before, and I want to just put it up again. So what I'm going to do now is I'm going to talk about this proofreading model, and when we did this in the morning, I put this term, a chemical potential X, as a delta mu, which for me sort of contained all the non-equilibrium driving forces. It was a chemical pump, and that was where all the forces were contained. So now we'll sort of go under the hood, and we'll sort of try to see how, they will try to sort of go into the mechanisms that a biophysical system can use in order to reduce errors. Okay, so again, the problem that we have at hand, and this is a very simplified description, obviously, is that you have a polymerase, and it can sort of add either the right monomer or the wrong monomer, and the error rate that you see in reality is much lower than the error rate that you have that you expect from equilibrium considerations. And today morning you sort of we sort of argued that if you want to go from here to here, you need to consume some energy, and the framework we wrote down allowed us to come up with reasonably good bounds on how, on what kind of energy you need to consume, but I didn't say anything about how the system should be structured so that it achieves this goal, how it can actually go from here to here, and that's what we're going to do right now. Okay, so that's the mechanistic picture that we're going to take, and we're going to unravel that right now. Okay, so let's sort of take the most naive zeroth order picture for how this polymer is going to grow. So I have this E over here, that's my enzyme state. So that's the enzyme that's unbound, there's nothing around. And I have these dots, and each of these dots represents for me an intermediate mesoscopic state. Okay, so imagine there's an enzyme floating around, and it's sort of, the enzyme has a DNA strand that is attached to it. And in this state, that's the state that I call E. This final state, which is E plus R, is where the enzyme has bound the right product to the growing template strand, and these dots here represent mesoscopic chemical states that the enzyme has to go through in order to go from here to here. Now, obviously, the number 4 is not sacrosanct. It's something that I just threw up here. I don't think it's clear what this number should be or what this number is, or whether there should be a number, whether it's just this to this. But it's just a useful representation to have in the back of your mind as we start thinking about these things. So I have this enzyme here. That's one mesoscopic state. This E plus R is the state where the enzyme has achieved the goal, and it has bound the right monomer, or it has done it has done the replication in the right way, and it has gone through these set of intermediate states. Okay, imagine that the y-axis denotes an energy. Okay, so this is, you start over here and you come over here, and in the process, if you want this to be a spontaneous process, the energy of this state has to be lower than the energy of this. Okay, so that's why I've sort of structured this like this. The y-axis represents an energy scale, and you sort of flow down in energy to come from here to here, which is to say that if that these chemical reactions are exothermic, they release energy, and so given a chance, the enzyme would indeed like to have bind something to the DNA. There might be hills and barriers as you come from here to here. I'm not talking a lot about them. Okay, so that's the right state, and as we discussed, thermodynamically there are other options. The enzyme can start over here, and it can sort of wind out, and it can come to the wrong state through another series of mesoscopic states, and that's what I call E plus W. And there's an energy difference between these two endpoints, and the probability of seeing errors, at least in equilibrium, is proportional to the exponential of this energy difference. KBT is my, T here is the ambient temperature of the body in this case, or the room temperature. Okay, so this is a very simple, diagrams of this sort are often used in statistical mechanics or in chemical physics, and it just sort of shows you a pathway. And so the question is, how do I go over this? How do I achieve an error rate that's lower than this number over here? This problem was first, I think, addressed by Hopfield Nino in close proximity, and without sort of going through a lot of history, let me sort of just sketch out the solution that they came up with. And the idea was essentially, and I'm not doing a great justice to how they thought about it and how they unpacked the whole thing, but the idea essentially is that they postulated that the enzyme or this enzyme plus the DNA strand, in addition to these pathways, and these are just standard energetic pathways, so these are the pathways that an equilibrium system would have, they postulated that there are additional pathways, and I've called them rescue. I don't think it was originally called rescue, but let's just go with this terminology for reasons that will become clear later on. And what happens in these pathways is, so let's say I start over here and I come over here, there is a finite probability or a finite rate that the system completely, the system wants to go back from here to this E state. Okay, and this sounds like a counterintuitive thing because you would want to flow down, you would want to come here, but bear with me. So the postulate is that you sort of start here, and if you happen to hop over here, there's a finite probability that there are rescue pathways that take you from here all the way back. And there are rescue pathways that adorn every set of mesoscopic states over here. Okay, and there are similar rescue pathways over here on this branch. And if you posit that the probability of the rescue or the rate of rescues is higher in the wrong states than the right states. Okay, so here I've multiplied the probability of these rescues by e to the power of delta kbt. Okay, so there's a rate of rescue over here. The rate of rescue in the wrong side is k rescue times e to the power of delta kbt. And now you can ask me how does this discrimination happen? I'll come to that in a second. But if you do indeed postulate that this happens, and if you were to write down for this picture the simplest set of mass balance like equations or master equations, and if you were to solve for the error probabilities, you'll actually see that the error rate in this process is called proofreading. And the word proofreading comes in because in some sense the intuition here is that the enzyme sort of sits and looks at the state that it has gone to, and in some sense it proofreads and it figures out, it sort of has a mechanism to take a step back, and if the proofreading process is efficient, it takes a step back mainly when it's along this chunk, and it lets the system flow through when it's along this chunk. And this delta that I've put in here, it's just an extra term that I've put in for now, is in some sense the discriminating ability of the enzyme or the proofreading ability of the enzyme, and that's what's going on over here. So if you were to take this network and if you were to solve for the steady state probabilities, and this is just a very simple exercise, I encourage you to do it. It's just a couple of lines or a page at best of algebra. What you will see is that the steady state probabilities, the steady state error rate in such a process is going to scale like the exponential. I put this number 2 here. It's actually it doesn't have to be 2. It actually can be. It's actually just an algebraic constant. For this specific thing, I think it is indeed 2, but it can be 3/2 or 4 or whatever you want. So you can tune this number, so you can go from this equilibrium thing to error rates that are much lower than what you expect in equilibrium by sort of adding these kinds of rescue pathways. Okay, and so there are many questions here. The first is why does it happen, or does it happen in the first place? And there have been beautiful sorts of experiments that have been done since this mechanism was proposed, where they tried to grow a DNA strand in which in a solution that only contained the wrong answers. Okay, so let's say I have a strand which is a pure strand, and I give the DNA polymerase only the wrong monomers. It turns out that the rate of growth is extremely slow. And that sort of reflects the fact that the system keeps proofreading and keeps going back. But if you only have the wrong answer, every so often it's going to sort of come down and give you the wrong result. If you do the opposite experiment where you start off and provide the DNA polymerase with only the right monomers always, then the rate of transport or the rate of translation or replication is really fast, and that again sort of confirms this picture. Okay, this discrimination ability between the right and the wrong, in the context of what one talked about, or he'll talk about tomorrow, one can call it a Maxwell demon like structure. It's able to use some information and distinguish between these two states. Although right now I don't have any mutual information or anything thrown in here, but there is some physical mechanism that allows the system to discriminate between, discriminate when it's going down the right pathway or the wrong pathway. Physically, one manifestation of this is if you actually have wrong base pairing, you have stresses that build up, and the enzyme has been known to backtrack whenever it goes over a wrong base pair. So there are physical mechanisms that are responsible for this thing. But on the whole, this general mechanism seems to provide this kind of result. It makes predictions that are testable. It predicts that if you only go along the right pathway, you'll go down really fast. If you only are forced to go down along the wrong pathway, you'll go very slowly. And this is sort of the mechanism that Hopfield and others have come up with, and this is now an accepted mechanism. So, yep.
Yeah, to get these estimations of bounds, or I don't know how to call them, for the error.
Yeah.
You have to. This is something that I never understood in this proofreading business. That you have to assume also things about the barriers. So if you are free to
yeah, exactly.
In the two pathways, so the energies by themselves
No, you're completely right. I haven't told you a lot about the kinds of kinetic constants that go in here, and depending on how you split your kinetic constants up, and depending on what goes into the barrier and how you choose these things, these numbers can change. And that's why I said that this is not really a firm number. It's for a particular mechanism. It turns out that you get 2, but in general you can sort of push this number, and what I'll show you in a few slides is that you can make it n. Where n is a very large number, and there are prefactors that sort of show up. But this basic structure of rescues and everything is sort of what motivated these kinds of ideas. But you're right. It's depending on how you structure your, depending on whether you have high barriers or if you put all the if your rates are completely dominated by the energies, you'll get different results. Okay, yeah. So that's definitely true. Okay, so I've talked for quite some time about proofreading and everything. Where when we started off, or when in today morning's talk, I spent quite some time talking about energy dissipation in this delta mu term. There's no delta mu like object over here. These are I have called these things rescue pathways. But if you were careful, you would notice that. Each time I have put a rescue pathway in some sense I've started or I have allowed for some kind of a circulation, right? So if the system starts over here and if it flows down, it can sort of come back up through this rescue pathway, and in some sense you can sort of set up currents. And this is something I alluded to at the beginning of my lecture today morning. I said that whenever you see circulation or whenever you see currents, you should think about non-equilibrium forces, and that's exactly what happens over here. So it turns out that these rescue pathways, or by having these rescue pathways, the system is actually driven out of equilibrium, and these loops or these circulations, these currents are actually supported by non-equilibrium energy consumption. So there needs to be a reservoir of some sort that actually supports this thing. In the absence of any such reservoir, such a process cannot be sustained, and we'll quickly revert back to this error rate. So there's a lot to take in, and what I'm going to do is I'm going to sort of boil everything down to a very simple 3-state model. Again, for the experts in the audience, this might be repetitive, but I think it's useful to introduce these ideas in the context of really simple things once, and then never bother about it for the rest of your life. I'm instead of having those many states, let me just consider the simplest one of the simplest models with a cycle. So let's say I have 3 states, A, B, C, and keeping in line with what I talked about in the proofreading slide before, A, B, C represent 3 internal chemical states of some kind of a molecule. Okay. And I've sort of drawn out these rates. So there are rates of transitions between these mesoscopic states. There's a rate of transition from A to B. There's a rate of transition from B to A, and so on and so forth. And since these are 3 mesoscopic states that the system has, they have some energies. And so I'm going to say EA, EB, and EC are the energies of these 3 states. The first question is, are these rates related to these energies, or is the whole thing coupled? If the system is in equilibrium, which is to say that there are no currents at all, it turns out that these energies are related intricately to the rates that I've put in here. So the kinetic processes that show up in an equilibrium system are governed to some extent by the energies of these states. Okay, so let's just go ahead and see it. So if I concentrate on this link, I can write down this very simple expression for the probability flux across this link, and that's basically the probability. So if I want to look at the number of particles flowing from A to B, okay, what do? I say let me look at the probability that I'm in state A. I'll multiply that by the rate to go from A to B. I have to subtract from this number the reverse flux, and the reverse flux is the probability that I'm in B, and I jump from B to A. Okay, so this is JAB. This is the current that flows along the link from A to B. And in equilibrium, we know that there are no currents, so that's a requirement. That's a requirement of equilibrium. And in equilibrium, we also know that the probabilities are given simply by the Boltzmann distribution. So the probability of being in state A is simply the exponential of minus EA divided by KBT. The probability of being in state B is proportional to e to the power of minus EB divided by KBT. And so the requirement that there are no currents in combination with the requirement that the probabilities have this Boltzmann form simply give you, simply constrain the ratios of rates along links. In particular, if I look again along the AB link, the ratio of the rate to go from A to B divided by the ratio of the rate to go from B to A is a ratio of probabilities. It's equal to PB over PA, and that's equal to the exponential of EA minus EB over KT. Okay, so the statement, and Juan mentioned this in this first lecture. The statement is called the statement of detailed balance. It tells you that there are, when this condition is satisfied, it tells you that the rates of transitions between various mesoscopic states, or the ratios of those rates of transitions, are determined entirely by the energies of the states themselves. When you have detailed balance, you have no currents, the system is in equilibrium, everything is constant, there are no flows of any sort, you just have these jumps which are immediately balanced out. And this is the condition, this is the constraint for all equilibrium systems. Ideas like fluctuation dissipation theorems and so on and so forth are built around the condition of detailed balance. So this is something that we all take for granted, and something that we use in molecular simulations, in analytical calculations, and this is the specific form. Again, this is just a very simple introduction. I just wanted to have it in here for people who haven't seen it before. Okay, so this is the condition of detailed balance, and these rates are sort of related to energies. Now, what happens if I have a system that has a current? Okay, so what happens if I have a system in which the system sort of circulates between these three states? Okay, so for some time it's in state A, then it goes to state B, and then it goes to state C, and so on and so forth. Okay, so if I have a system that has a current, clearly this condition can't be satisfied, because this condition implies that J is equal to 0. So if J is not equal to 0, the ratio of these rates is no longer equal to the exponential of the energy difference between those two states. So detailed balance is violated, and in some sense I can use this violation of detailed balance. So now that this ratio is no longer equal to EA minus EB, I can use the offset, and I'm again going to use delta. I keep using this the energy delta, and I can say that KAB over KBA is e to the power of EA minus EB plus some offset term delta that sort of captures the detailed balance violation. Okay, so what's going on over here? A simple physical way to sort of think about delta is to sort of assume that whenever you go from A to B, in addition to the energy that you gain, by changing your state, you also have, this any jump from A to B is coupled to a non-equilibrium process in which fuel is burned, or a pump is turned, or something, or some kind of energy is input, and if you couple any jump from A to B with a process which burns fuel, you'll get this condition, and you can interpret delta as the chemical fuel that sort of drives the system from A to B, or it's the chemical cost that you need to pay to drive the system from A to B. And if you sort of unwrap the whole thing or invert the whole thing, if I give you a set of rates to go between states, you can take the log of those two rates, and you can interpret the log of those two rates as the total energy cost to go between those two states. And there are conditions under which this total energy cost has some external fuel constraints that are built into it. Okay, so that's what happens if you have currents. This KAB over KBA is no longer equal to the exponential of energy differences. You have to add this extra term here, delta, and this delta sort of tells you something about how much energy you need to consume or provide in order to sort of sustain these kinds of cycles. So this picture allows us to interpret what happens in the proofreading model. Before going further, to support this J, we need to sort of consume a chemical energy delta. This delta is in this case, so if I take the product of the current and the chemical energy delta, it's basically equal to the product of J and the log of KAB over KBA, and I define the product of the flux times this quantity, which I call a chemical affinity or a chemical potential flux, or you can call it chemical potential. I define the product of these two as something like a power. Right, so this has some units of energy, this has some units of flux, and if I take the product, I get something that looks like a power. And indeed, if you were to do if you were to go through all these calculations, you find that if for a network of this sort, the amount of power consumed in order to support a current J is proportional to J times the log of the ratio of the rates taken in the clockwise direction divided by the ratio of the rates in the counterclockwise direction. Okay, and so I've skipped a few steps here to go from here to here. It's a very simple calculation, but in the context of this simple one-state model, the amount of power that you need to consume depends on J, and this J depends on all these constants. It's not a closed solution, but it's just it's an expression that tells you what goes where. The amount of power that you require is proportional to the current that's driven through the network times the log of the ratio of the product of rates taken in the clockwise and the counterclockwise sense. Now this is for the simple three-state network. It turned yep. Yeah.
They're same.
Yeah, exactly. So I'll come to that. Yeah. So if the condition of detail balance is satisfied, then KAB over KBA is going to be e to the power of EA minus EB. This is going to be e to the power of EB minus EC. This is going to be e to the power of EC minus EA, and this whole thing is simply 1.
No, you're saying like Kolmogorov.
Sorry, what?
Kolmogorov.
No, so I don't even want to go there right now. Yeah, that's the interpretation that all this is based on. I don't really want to go there. I'm just saying, one can take the log of these rates, and I can think of that as an energy flux or an energy source. This is a consistent interpretation for the power that's required to sustain fluxes in a non-equilibrium network. Okay, so that's what I'm trying to say. And if the system is in equilibrium, then KAB or KBA is e to the power of delta EAB. And similarly, these are also all of these are energy differences. And so if you go clockwise and go counterclockwise, you end up with the same energy difference. And so this term drops out, and the power required to sustain something in equilibrium is 0, which is consistent with our intuition. And if you go out of equilibrium, if this ratio of rates is different from this ratio of rates, then you require a non-zero power in order to sustain currents. What I haven't shown you here, and this is sort of related to what Anupam asked right now, I think, is that this function has many nice properties. It turns out that this function is always non-negative. You can show that in a few lines. The proofs for non-negativity are very similar to the kind of proofs you would do when you think about the non-negativity of the relative entropy. So you can show that this is non-negative. This is also related intricately to if you think of this whole thing as a dynamical operator, terms like this are intricately related to Lyapunov exponents that show up in the analysis of the dynamical operator that decides the dynamics of the system. Okay, so I'm not going to go into those things. I just wanted to use this as an excuse to sort of point out that if I were to give you a chemical reaction rate network, you can take the rates and you can start thinking about ideas like work. Okay, so because usually the kind of things that one wrote on today morning, work there has a very physical meaning. It was I take a Hamiltonian and I change a parameter, or I push a piston, and in that case we are used to thinking about FDX kind of work. Even in the context of these biochemical reaction networks, work has one can similarly define work. It has a very chemical meaning, and it's usually expressed in terms of structures like this, where the log of the ratio of the rates provides some kind of an affinity, and J is a flux in response to the affinity. Okay, and this is the kind of structure that one encounters when one wants to do thermodynamics with these kinds of reaction networks. Okay, so now that I have all these things set up, I can basically claim that whenever you have these kinds of pathways and additional pathways, these additional pathways necessarily require non-equilibrium energy consumption, because the way I've drawn it, it'll turn out, or it's necessary that the ratio of counterclockwise fluxes is different from the ratio of clockwise fluxes, because if that's not the case, the system will just be in equilibrium. There won't be any enhancement of the, or any reduction of the error. And these non-equilibrium fluxes end up supporting this pathway. These non-equilibrium fluxes provide the delta mu that I talked about in the first lecture today morning. And if I give you all the rates, and this is a useful exercise to do, and this is something that is done in the additional reading paper 2 by Mehta and Schwab. So they take networks of this sort for a different purpose, and they work out entropy production for those networks and show that it is intricately related to an information processing purpose of the network. Okay, so you can take these rates and you can use them to come up with thermodynamic values for power and so on and so forth, and you can see how the energy consumption is crucial in order to sort of take you from an error rate that is on the order of e to the power of minus delta kBt to an error rate that's is like e to the power of minus 2, or in general a larger exponent times delta kBt. Okay, so the delta mu from today morning is sort of contained in these non-equilibrium cycles. So these are basically random walkers. It's just that I have decorated them and have made them slightly more complicated. But basically, it's the same kind of the machinery you would use to analyze them is exactly the same machinery you would use to analyze random walkers. I have just thrown in this new term, which is called energy consumption. Okay. Now this might look like something that I've just constructed, but as I said, this term has many interesting properties, and we'll see some of them as we go along. It's intricately related to information processing for the reasons that we discussed today morning. The delta mu term sort of is captured here. It's related to things like Lyapunov exponent for these dynamical operators. We'll also see that this energy consumption rate, it constrains fluctuations of various quantities, and it allows us to do a lot of cool things. Even it so happens that the structure that's sort of built into this places, gives this quantity that we have constructed, which is this rather innocent-looking product. This product has many properties that that allow us to. So once we estimate this product, we can play a lot of cool games and talk about how fluctuations of various other quantities are constrained by fluctuations of this product. Okay, so that's it has a significance that sort of is hidden and which I haven't done justice to yet, but I will talk about it as I go along. Yep. Sorry, what? Yeah, exactly. So the energy consumption will be different in the two parts, and what that's going to be determined by is what are the fluxes that go into each of these things, right? Because in this case, if my rate of rescue is really high, right? If k rescue times e to the power delta is very high, and the system is almost always just over here, then it can actually consume less energy in the wrong state, but it can consume more energy in the right state because it sort of flows down more. So it's not a priori clear how this thing plays out, but the answer is yes. So the energy consumption along these two pathways is going to be different, but it's not clear that the energy consumption along this pathway is necessarily higher or greater than this pathway for now. Okay. So in general, this formula is always valid. I have written down this formula for the case of one loop. Okay, if you have a system with many different loops, you need to sort of take the effectively, and I'm sort of one can do this thing more carefully, but effectively one needs to take the current in each independent loop and multiply it by the log of this ratio in each independent loop. So you need to take your big network, decompose it into independent loops, think about the current in each independent loop, and think about products of this sort in each independent loop. Okay, and that's how you would get this thing. So the sum of all those things is going to be a total entropy production rate, and it has all the properties that I just mentioned. It's going to be non-negative. It's actually a convex function. Yeah, it's got all the properties that this object has, and it's easier to see here. Okay, all right. Anything else? Okay. Okay, so it's at this point, I do want to sort of ask you to pay attention to this additional reading too, because this paper uses these ideas, and there are results showing you how this dissipation or energy consumption controls information processing properties of this particular network that they consider here. Okay, so we introduced this, and before moving ahead, and now that we're talking about random walkers and energy consumption and things like that, let me spend some time, illustrating a few more properties of this energy consumption or this energy dissipation quantity or entropy production quantity, and so let me sort of add some more complexity here. So there are a few more states. So this is a bigger loop. And this is basically a model for some kind of a asymmetric random walker, or this could be a model for a system or a molecule that has many different chemical states, and you have rates of transitions between the various chemical states. For simplicity, I'm saying that all the rates of transitions in the clockwise sense are kCW, and all the rates of transitions in the counterclockwise sense are kCCW. Okay, that's my setup. The system has obviously a current, which is going to be related to the difference of kCW and kCCW. Maybe there should be a proportionality sign. If these two rates are different, you're going to set up fluxes in the system. This is not the current J. This is just a parameter V. I think I'm missing a factor here. This is just a parameter V that sort of determines if there's going to be a current in the system. And as we discussed, needs to consume energy in order to maintain this random walk. One can define by analogy from what we had before, and I think I'm missing some factors here, but this would basically be right. The rate or the power required to maintain this cycling whenever the rates are mismatched is equal to V, which is this tells you how fast the system is flowing around, times the log of kCW divided by kCCW. Okay, so this is exactly the formula that I had before with KAB, KBC, and everything. Right now, I just have these two rates, so it gets simplified, and this is my total energy consumption rate for the system. So this is defined. I should put angle brackets here. This is the average energy consumption rate. What I can do is I can do something more specific. I can actually go ahead and look at. I can consider the energy that the system consumes as it marches along a trajectory. So whenever the particle goes from here to here, I add a log of k CW divided by kCCW, and whenever the particle goes from here to here, or it makes a counterclockwise jump, I add a log of kCCW divided by log of kCW. Okay, so in some sense, I can sort of try to account for the energy gained or lost in each step, and I can keep adding them. On average, if I look at the average amount of energy, I'll get this quantity, but I can consider these things on a trajectory basis, and I can go ahead and tabulate the whole thing. So if you were to tabulate this whole thing, and if you were to plot histograms of the probability of observing a certain energy consumption rate along a trajectory, and compare it with the histogram of the probability of observing the negative of that energy consumption rate. So these are, I'm sorry about the notation, this should be an angle bracket over here because this is an average quantity. These are fluctuating quantities over here. It turns out that the ratio of these two probabilities is equal to, in the long time limit, the exponential of W dot times t, where t is the time that you observe the system over. So this is again a so-called fluctuation theorem, which is very similar to what Juan talked about today morning. And this version of the fluctuation theorem for processes on such chemical reaction networks was proven by, among others, by Leibowitz and Spohn. This beautiful paper from 1998 or 1999. I might have messed that up. But this is a different version of the fluctuation theorem in the kind of analysis that one had. We had pistons and gases and changing parameters. Over here, the nature of work is slightly more involved. It's sort of built in to the dynamics of the system. Whenever the system makes a jump from here to here, it either consumes or releases energy depending on how these rates are structured. But it turns out that even this accounting of work ends up giving you results that look remarkably similar, and in fact are very closely related to the results that one presented today morning. And this is another version of the fluctuation theorem. Now, today morning, Anupam brought up this notion of a cumulant generating function or a large deviation function. So if I take this probability distribution P and consider its appropriate effectively Legendre transform, I can go to a basis, I can go to a different basis and I can construct the cumulants of this fluctuating quantity W, and it turns out that this fluctuation theorem it reflects a fundamental symmetry in that cumulant generating function. Again, this last thing was for experts who sort of know and think about this, but the point I'm trying to make is that this quantity satisfies many symmetries that are not immediately apparent when you first write it down, but the symmetry shows up. In this shows up in almost all classes of driven non-equilibrium systems. So there's one thing that one mentioned in the context of particles in which you change parameters, but it also shows up over here. Just that one needs to think carefully about how work comes in here. Okay. So that's one property. The second property is something that I used today morning to derive my bounds, and I'm just going to talk about two. I'm not doing justice to the vast amount of literature that's out there. So if I go back to this V, which is my drift-like term, this is the difference of the clockwise rates and the counterclockwise rates. I can also construct something. I can also construct the arithmetic mean, and this sort of measures, in some sense, the diffusion of the system. It sort of tells you how the system diffuses around and loses track of its surroundings. and I can construct this quantity, which is the ratio of this diffusion to the square of the drift. Okay, so this sort of is a measure of fluctuations in the system. Okay? And it turns out, and this is what I used today morning in my to construct the bound that I described. It turns out, and this is a beautiful result proven by Barato and Seifert for the first time. It turns out that the entropy production, and I'm sorry, I've changed the convention. So this my sigma here denotes energy consumption now. I used W dot before. Sigma here is energy consumption. So it turns out that the sigma not only has to be positive, which is what I described before, it not only satisfies the fluctuation theorem, it also has to, the product of sigma and this quantity epsilon squared, which sort of tells you something about fluctuations in the system, this product, it turns out, has to exceed twice kBt. Okay, and this it has a flavor of an uncertainty-like principle. This relation is not contained in the fluctuation theorem that I showed you before. It's related to the fluctuation dissipation relation, and I view this as a non-equilibrium generalization of the fluctuation dissipation relation. But again, it sort of tells you something about how the sigma, in some sense, controls fluctuations of various different quantities in the system. So this sigma measures energy consumption, this epsilon measures fluctuations in transport properties, and this result shows you, and this result is derived from an analysis of using large deviation principles and so on and so forth. And I can talk to you offline if you're interested about that. But it shows you that this quantity sigma, even though it's rather innocent looking, it actually controls fluctations of quantities that you wouldn't immediately associate with energy consumption. So simple transport properties, it turns out, are also controlled by the energy consumption rate sigma. And the reason I'm harping about this for such a long time is in some sense to justify the interest or the recent interest in thinking about things like trade-offs between energy, speed, and accuracy. Relations of this sort do indeed sort of suggest that thinking about energy consumption rate can actually be a fruitful quantity because it does indeed constrain fluctuations of various seemingly unrelated quantities. Okay, so this result was first postulated in a paper by Barato and Seifert, and it was proven later in a PRL by these people. Okay. All right, so this is sort of a detour, but I wanted to use this as an excuse to talk about energy consumption and its properties and so on and so forth, because it's useful to, it sort of serves, it gives you a basis for why we spend or why we are spending so much time thinking about this quantity. It just turns out that it has these symmetries which constrain fluctuations of various unrelated quantities. And this is sort of, and this relation was the basis for the relation that I wrote down today morning, where where we could use this relation to estimate patterns in self-assembly. This is actually a useful exercise for it's useful to sort of do this on your own. You can consider a random walker with rates KCW and CCW, and you can actually derive this. It's just an algebraic exercise, but I would highly recommend that you do this. You'll waste a few pieces of paper, but you'll be better for it. All right. So now that I've spent all this time talking about cycles and networks, if I go back to the thing I talked about today morning, which was this huge growing polymer, there is actually a way to take this growing polymer and map it onto an effective random walk-like system. So there are some approximations that go into this mapping. But if you were to do that, and effectively the essential element here is you look at the edge, and if the edge has a defect state, you call it a D. If the edge has a state that has a bond between like pairs, you call it an S. And in some sense, the dynamics of the system are the dynamics of a random walker in which you hop between D and S with some specific rates. So the mapping is somewhat involved, but you can do it. It's not very complicated. And if you were to do this mapping, and if you were to use this prescription that I just gave you for entropy production, where you take the log of the kinetic rates and the currents, you actually can show in a couple of lines that for this system and for this effective system, the entropy production rate, and sorry, I've used sigma again instead of W dot, is indeed equal to what I wrote down today morning, and this relative entropy-like term shows up naturally if you go through such a construction. And this should again help you for one of the problems that I listed in the handout that's there on the website. So yeah, again, so this kind of a framework is very useful. You can start here where it's easy to account for things like heat. You can go to this effective model where all the accounting for heat is sort of hidden in the kinetic constants of the network. But if you use the framework that I provided in the last few slides, you can a few lines you can show that the entropy production rate is indeed equal to the answer that Bennett and others arrived from a very different set of considerations. So it's again a useful exercise to do, and then you can also see how you can apply ideas like this uncertainty principle in the context of polymer growth or self-assembly. Okay, so now that I have all these things out of the way, let's go back to proofreading, and now sort of I want to think about this problem more generally. So I had this simple 4-state model, and I said that you can solve it, and I said that you can write out detailed solutions for this 4-state model. But in general, we expect our networks or our systems to be much more complicated. We expect that there are going to be many different mesoscopic states. We expect that there will be many loops that the system can go through. It's not just this one loop or this a few set of loops. And in some sense, the question we're trying to ask, or sort of searching for design principles that allow the enzyme to localize its probability or to localize all the likelihood around the right product, and to localize all the likelihood away from the wrong product. Okay, and so how do we, are there general principles that allow us to do this kind of that allow us to achieve this kind of discrimination? Obviously, I can go in and we can try to solve it model by model and see how energy consumption plays a role in this discrimination, or we can talk about how the kinetic structure of the rates here gets you this. But the question that I want to sort of try to motivate now is, are there general principles that seem to govern, or that we can use in order to anticipate these kinds of behaviors? And to make life more complicated, usually in biological systems you have there is noise, there's disorder in the kinetics, and that's because of thermal fluctuations, and so the whole thing is usually a mess. Okay, and what we want to try to do is we want to see if there is any way for us to try and come up with general design principles that lets us understand how we can actually achieve discrimination in a broader context. Okay, so In order to do this thing, we can start off with a particular reaction rate network, and let's say you give me the rates of transitions between all the mesoscopic states. So these are the rates that I wrote down. I can construct a dynamical equation where I evolve the probability of the system, where I evolve the probability that the system is there in any of these states according to what's called a master equation. So this P here is a vector that has information about the likelihood or the probability that the system spends time in any of these mesoscopic states. W is a matrix that tells that has information about the rates of transitions. And so I can put all this together. I can write down a master equation, and I can try to solve it, and I can try to see what happens. I can look at the steady states. But this is a horrendously complicated process, and one has to do this on a case-by-case basis. If the system were in equilibrium, I could simply use the energies of all these states, and I could predict what would happen. But the system is not in equilibrium, and in that case, what can we say about the set of processes? Does one always expect localization like this? Around the right product, localization away from the wrong product? Is there a way to go between these two regimes in a very general manner? So what I'll do now is I will talk about an analogy between the proofreading system and a different class of biophysical systems in which these kinds of transitions or these kind of steady states have been experimentally observed. And I'm doing this just to sort of motivate the fact that even though we have moved away from the Boltzmann distribution, we don't have a very clear idea of what a landscape looks like. There seem to be some notion of universality, or there seem to be some notion of design principles even in these classes of systems. So what I'm going to do is, and this is why I use the word rescues, I'm going to use an analogy between the circuit that I drew out, which is for proofreading, and a very, very different biophysical system, and that's microtubule. So microtubules are there in the cells. They sort of determine cellular architecture. They transmit forces. They transmit forces in cells, and very generically, the dynamical behavior of microtubules can be captured by the schematic over here. This microtubule, it turns out, has two kinds of monomers. It has a monomer which is bound to GTP, and there's another kind of monomer which is bound to GDP. So these are two different nucleotides. So this is a phosphorylated version of this nucleotide. And it turns out that if you have the blue GTP bound monomers, the system can actually grow. But if this cap falls off, the system basically shrinks and it sort of falls off. Okay, so this is a very different dynamical process. But notice I've used the words rescue and catastrophe again over here. So if you were to write down the dynamical models for the system, and this is also a driven non-equilibrium system, because if you want to go from this state to this state, you have to consume energy. So you put in cycles in this case. And if you were to look experimentally at the phase behavior of the system, you actually find two very interesting regimes. You find one regime where the probability distribution of lengths is completely bounded. It sort of fluctuates about 0. And there's another regime where the probability distribution of lengths is completely unbounded, and it keeps growing up. Okay. And these two regimes are very useful or interesting regimes, because if we could take a kinetic proofreading network, okay, and if we could ensure that the right product is in this unbounded regime, then the system, whenever it sort of flows down the right product, will immediately come out. It will have very high transport properties. And if you could ensure that the wrong product is in the bounded regime, then the system, whenever it goes the wrong side, it sort of immediately gets pushed back, and you have very high discrimination. Okay, so this is a very different system. It has a very different kinetic scheme. But I wanted to point this out because it seems to suggest that by using non-equilibrium forces, I can get much more than just these steady states. I can actually No, it's actually. I don't have a good, I don't know what the exact distribution is. I know that the bounded regime is exponential. I don't know what the, yeah. So I mean, I can imagine a regime where it just sort of grows linearly with time. There is a transition point where you go from here to here, and at that point you can have interesting statistics. But if you're deep in the unbounded regime, you're going to grow linearly, and if you're deep in the bounded regime, you're going to have an exponential behavior. And it turns out that you can actually tune this system to lie, in a very sharp manner to go from bounded to unbounded. So this almost looks like a phase transition, but in a dynamical context. And then at that point you get hysteresis. You can get more interesting behavior at that point. But I don't know what the behavior is over there. But deep in the unbounded regime, you sort of keep growing. I think microtubules typically exist closer to the bounded regime. I think they don't just keep shooting off. So the regime they exist in, and I'll come to that in a few seconds, is they exist. Oh, here it is. Is they exist in a regime that is close to the bounded unbounded transition, and that way they can be bound, and they don't have to consume a lot of energy, because whenever you transport something throughout the network, you consume a lot of energy. But by being close to the transition, every so often they can shoot off, and they can. So this is about food, but microtubules sort of. The microtubules use similar mechanisms to find cargo in cells. E. coli uses this mechanism, or uses this run and tumble kind of motion to find food. And so you can have this unbounded motion, and then you can have these catastrophes that bring you down. And having a combination of these two allows you to search very efficiently for food. And that's the reason why I use the words rescue and catastrophe. And all this is work by this part is work is one of the references I sent out before the lecture started. There's a PNAS paper by Arvind Murugan, David Hughes, and Stan Liebler, which sort of made this connection between microtubules and proofreading, and I find that very instructive and useful to think about information processing ideas in biology. Okay, so the reason I went on this excursion with microtubules is just to point out that this system, which has structures that we can possibly use, has these very interesting regimes, and so then these regimes can achieve, in this case, can allow the system to search very efficiently. And if this regime was allowed in the proofreading system, then it would help the right products get translated really fast, and it would help the wrong products. It will ensure that the wrong products are localized and and don't get translated at all. And it's sort of a very efficient way to set up the system, because only the right gets done and the wrong doesn't get done. So you achieve a balance between transport, or between speed and accuracy, by doing this kind of a thing. So it turns out that if we go back to the kind of kinetic proofreading networks that I was drawing out, we can regard these arrows as rescue pathways effectively. And again, for a large class of networks, and you can sort of you can do a generating function approach, look at the transport properties of such networks, and what you find is that indeed for these kinetic networks, there are many different dynamical regimes that actually emerge. So there's one dynamical regime where the distribution or the system is almost entirely localized at the reactants. Okay. Yep. Sorry, what?
How does the RMS, what is the variation?
Yeah. Okay. So it turns out it depends on where you are actually, and I'll put that. So so this is so in this case everything is bounded, right? So it's nothing much is happening. In this case, you're almost like ballistic motion. So you have small fluctuations about it, but nothing much is happening. In between these two, you actually get large fluctuations, and that's what is called dynamical instability, because you sort of go between regions where you are forced back, and regions where you sort of just shoot off, and you come back after a long time. Okay, and so in that region you really have large fluctuations, and it looks like a phase transition, but in a very different context. And again, in a few minutes I'll sort of talk about how dissipation seems to control these emergent features. So I'm moving really away from information theory, right, for a few minutes, but just talking about how these ideas which we used today morning to do something else can be used in other creative ways to achieve information processing function. Okay, so if you go ahead and look at these kind of networks, and at this point you still have to you can consider a large class of these networks, and if you solve for the transport properties of these networks by looking at the first passage times and other kinetic processes, you indeed see that there are these regimes. You see regimes where the probability distribution is localized at the reactants. There are regimes where the probability distribution is delocalized and sort of it spans the entire network, and there are other regimes where the probability distribution is localized on the products part. When you have localization here, the error rate scales like some prefactor alpha times n, where n is the number of nodes in the system or the extent of the system. And in some sense, we want to have an interplay between this region here and this region here. We want this region for the right products, because the system can quickly go through and generate the right kind of bonds, and we want this region for the wrong products, so that the system doesn't actually form the wrong products. It's just that this delocalized region consumes a lot of energy because you keep transporting things in, and so having an idea of these regimes now allows us to, we can now start asking questions or answer questions about trade-offs between energy consumption and efficiency. This regime you consume a lot of energy, but you get a trade-off for high transport. In this regime, you consume very little energy, but you get very low transport, and so you can sort of start playing games for. You can start asking questions about how you want to optimize your features. The important point I wanted to make with this graph is simply that even in these kinetic networks, it turns out that there are these three regimes that seem to be separated by something that looks like a phase transition, or it's not really a phase transition. There are sharp features that separate these three regimes, and you can hope to sit at boundaries of phase transitions, and you can hope to utilize non-equilibrium fluctuations in the most efficient manner to get the best information processing function. So in other words, if I take a dynamical model which has none of these non-equilibrium fluxes or which is very small, typically if you look at the trade-off between error and dissipation, you'll get a curve which is very boring. This is just a standard trade-off curve. But if you have models which have the so-called dynamical phase coexistence, you actually have two different branches. You have a branch where you get a large gain in transport properties at a small cost, or you can get a small gain at a large cost. And so this is the high delocalized regime, and this is the localized regime. And you might want to sit somewhere in between these two regimes in order to efficiently use energy and reduce errors. So the point of all this is just to show or to point out that these simple non-equilibrium biochemical reaction rate networks, if you were to look more closely at the kinds of structures that emerge, it looks like you get structures that resemble phase transitions. Dissipation still plays a very important role. It controls a lot of this behavior, but there are many different regimes that you can go between. And if you want to think carefully about information processing in these systems, one needs to pay close attention to whether these regimes exist and whether there are sweet spots that you can find where you can get the most bang for buck. Okay? And to again harp on the point that the dissipation is important for systems like this. If I were to plot a distribution of energy consumption rates, you almost always find you'll find curves that look a lot like the kind of landscapes you draw out for equilibrium, for equilibrium liquids and so on and so forth. You see that if I were to plot the log of the probability of sampling a certain dissipation rate, I get a distribution that looks like the blue curve. It has a very fat tail that represents or that seems to presage some kind of a phase transition, and this information is again contained in the fluctuations of energy consumption rate or dissipation rate. And by tuning the system to the right phase transition point, you can hope to achieve the function you want. So that's proofreading. In this, there's another example that I wanted to talk about in the last few minutes that I have, and in this example, this is related to how cells sense concentrations and respond to concentration changes. Again, I won't be doing a very thorough job of talking about everything here. This is another workhorse model that people use to think about information processing and robustness. And I'll quickly throw out a few features that this has. And again, there is a connection here between robustness and information processing in this model and energy consumption rates. Okay, so I have here my a caricature of a cell. This is an E. coli cell, and if you look at how an E. coli moves, it has this run and tumble motion. It sort of tumbles around and it runs, and it tumbles around and it runs. And this is very useful because, as we discussed a few slides ago, a motion like this is very useful for search. Because you tumble around, you're effectively localized here, and then you run, you sample a large region, and then you again search a small region, and so on and so forth. So it's very useful to have this property when you're searching. And this run and tumble motion is controlled by the activity of some proteins that are up here at the head of the cell, and I'm using the word head in a very colloquial sense. And so if the system is in an active state, you have tumbling. If the system is in an inactive state, A equal to 0, you have runs, and so on and so forth. So there's some proteins over here that I can assign an activity to these proteins, and depending on how these proteins, depending on the state of the proteins, you have this run and tumble motion. Now, if I were to pulse this, if I were to show the cell, or if I were to pulse the cell with a certain chemotractant concentration as a function of time, so this is, I sort of start off with the chemotractant concentration, it's constant, and then it increases, becomes constant again, and then it changes, it becomes constant again, changes, it becomes constant again. In some sense, what I want the cell to do is I want the cell to act like a filter that only senses gradients in time. I don't want it to sense these absolutes, changes in the absolute values, because this is just a point where everything is uniform again, and it should go back to its default run and tumble behavior so that it searches the most efficient way. But I wanted to sense these gradients. I want the cell to perform a very, very specific signal processing goal. But everything here, all these objects are governed by statistical mechanics or laws of statistical mechanics. So the question is, how does it happen? How can the cell take the signal and efficiently only sense these points, and in some sense adapt to these new concentrations? So when the chemotractant concentration hits a constant again, I want the cell to go back to how it was running and tumbling. But when it senses a large spike in concentration, I want the cell to either go towards it with focus, or to run away from it or tumble away from it if it's a negative chemotractant, or if it's something that's not favorable. Okay. So in some sense, I want the activity of this chemical complex up here to have a behavior that looks like this. Over here I have a constant chemotractant concentration. The activity is a constant. This is a number that I've just chosen as 0.5, and in this 0.5, the system is running and tumbling. It's searching very efficiently. Over here, it sees a large spike in favorable conditions, and so the activity drops to 0. It runs towards this large spike. And once the system sees that there are no more transients, I want the activity to come back to the 0.5 value where it can search very efficiently. And over here, if it sees a large spike going the other way, you want it to tumble away and take evasive action. But again, you want it to come back to its steady state. Okay, and so in some sense, this protein network over here does a remarkable job. It's able to measure these concentrations and is able to achieve this very, very cool signal processing like function. And this is all this is noisy. There are the numbers here on the order of hundreds of molecules, but it's able to achieve this activity or this behavior with remarkable precision. This whole process is called adaptation, because the cell adapts to concentrations, and it turns out this process is very robust to changes in many different conditions, and the state is maintained in a robust manner. Okay. So the question, ignore this for now. So the question I want to try to ask is how does this happen? What are the physical principles that are responsible that led the cell achieve this kind of a controlled state? And in order to sort of, yep.
So even if you have uniform gradient of chemicals, would it show?
So if you have a uniform gradient in time, Yeah, so it turns out, I mean, this is obviously an idealized thing, but if you, in this idealized model, if you had a uniform gradient and if you kept showing it things, then it would keep moving the right way. Yeah, so you'll have tumbling at a slower rate, but it'll keep till it hits saturation, it'll keep sort of following the signal. Okay, so that's sort of built into this kind of a thing. I don't know about. I think even in a in a real biophysical system or in a real experiment, I think you can saturate things, and at some point it'll stop running and it'll just tumble. I think that's what will happen. But in the context of what I showed you, this thing will just keep moving. Okay. Yeah. So the results I showed, the graphs I showed you here were quick. Things that I cooked up for this lecture. It was just a few simple Mathematica code that I use for that. Okay, so there's this network of proteins, and I've represented this network in this manner over here. This picture has no real significance; it's just a qualitative picture. So this network of proteins is able to sort of do this function. People have actually looked carefully at the internal state of the proteins, and they've actually found that as this activity is maintained as a function of the signal strength, it turns out that the protein uses an internal chemical variable, and this variable is the methylation state of the proteins, and so it uses this methylation state, or this red line, as some kind of an effective control variable. Okay, in some sense, to sort of get the activity back to this set point. This is a graph that I've generated here. This S is in some sense it represents the chemical signal or the chemotactin signal. There are points beyond which the system fails to adapt. It doesn't do its job. But there's a large region where it adapts very efficiently. That's the activity, and it uses this internal chemical variable or this internal chemical modification scheme to achieve this information processing function. So unlike proofreading, where we were thinking about errors in binding, the error here is a loss in adaptation, or a loss or a state in which the activity goes away from 0.5, and it turns out that the cell somehow uses this internal variable to achieve very, very efficient adaptation. And again, even in this case, people have now figured out, or have now demonstrated, that if you were to look at the state space of activity and methylation for this protein complex, you actually again find cycles. The system sort of goes through some kinds of cycles. So there are indeed non-equilibrium dynamics that are at play over here, and these non-equilibrium dynamics end up maintaining the steady state of the system even in the presence of large thermal noise and other such effects. So what I'm going to do now is I won't do the math. I'll just sort of point out a few things that emerge if one writes down a simple chemical reaction state network for this protein complex. So what I have here is on the y-axis I have activity, active and inactive states. On the x-axis I have methylation levels, and this resembles the kind of networks I wrote down to proofreading. They have a similar structure. In proofreading, we would have called these pathways rescue pathways. Over here, these are just pathways that the cell uses to go between various states. And there are cycles over here that just denote the fact that the system consumes energy in order to achieve this state. And so again, one can sort of go ahead and write down the so-called master equation, where you write down probability. You can write down, you can construct fluxes and so on and so forth. And the question is, how does the system achieve adaptation even in these noisy conditions? And it uses this methylation internal variable to achieve this. How does all this happen? What are the requirements for this to happen? Now, this is a question that one can give a whole lecture on. I don't have that much time right now. If you take a network of this sort, and if you compute the energy consumption rate using the prescription from the beginning of this lecture, what you actually see something really remarkable. You actually see that the energy consumption rate, or W dot, constrains. So this quantity here, the log of epsilon naught over epsilon, is epsilon here happens to be the difference in the activity. So epsilon is over here is defined as the mean activity minus the activity that you desire. So in our case, it's 0.5, and so epsilon sort of picks out how far the system is from the set point that it should be at. And it turns out that this energy consumption rate is intricately connected to the logarithm of 1 over epsilon. So in other words, if I want a system that's extremely accurate in terms of adaptation, if I want a system that adapts really efficiently and always maintains the set point of 0.5, it has to necessarily consume a very, very high amount of energy in order to do this thing. And all this again emerges if you use the formulation I gave in the beginning of this lecture and go ahead and compute entropy production rates. You actually see scaling laws of the sort that emerge, and this seems to be reasonably robust for this class of models. You see that the error rate, which is again given by epsilon, does indeed depend on how much energy you consume, and it's you sort of have this logarithmic or exponential dependence, which seems to constrain which seems to sort of constrain the error rates. Even in the context of proofreading, if you were to go ahead and solve the models that I showed you before, you'll find similar scaling features, not exactly the same form, but they have very, very similar constructs or very, very similar features. So the question that I'll end my set of lectures with is so in everything I've done in this last lecture, I've sort of tried to point to big connections. I've not solved any problem per se, but I've taken these 3 different problems. I started off with proofreading, and then I went to microtubule growth and the instabilities in microtubule growth, and then finally adaptation. In each of these problems, you have energy dissipation. The system consumes energy. Each of those problems uses energy consumption to achieve a very specific function. Okay, and it uses it by sort of going through dynamical phases and by doing other things. And it looks like there are hints of universality here, right? So it looks like you can take all these different systems, and even though they have very different specific ideas or objects built in, it looks like there are some localized states or some steady states that emerge that have very, very similar features, and all this seems to suggest that one can actually march towards thinking about building robust localized modes into networks of the sort. If the system were in equilibrium, our answer would be very simple. This would just be the Boltzmann distribution, but this is non-equilibrium. But the examples I have sort of shown you hopefully have at least made it plausible that one can maybe come up with a framework that allows us to think about how very specific function can be programmed in to networks of this sort. So at this point, I want to sort of point to a couple of the questions that I had in the handout. One of the questions, I think it's a third question, is on is a really beautiful paper which uses tools from signal processing and applies them to biochemical networks, and it actually shows that biochemical networks can perform as optimal filters. And so the way they're set up, it turns out they are set up in a manner that optimally suppresses noise and performs its function. As an exercise, I think it's very useful, and I'll be around tomorrow to talk about this if you're interested more. It's very, very useful to think about how ideas like that, which sort of bring in signal processing, are related to what I mentioned today and what I mentioned in the lecture yesterday related to fluctuation dissipation like ideas. And I think that's it's something that I'm very interested in, and I think it's an avenue for some good biophysical work in the future. So let me end by sort of giving a partial answer to this question: can robust modes be programmed? And I will, this last 3 slides will be very technical. I'll just sort of give you a broad overview of where I am heading. This is sort of work that I'm getting interested in right now, and just to sort of tell you how I like to think about these things. I won't do any justice to the details here, but I'll be happy to talk to you offline. What we have sort of found is that there is actually a way to take these classical stochastic master equations and to map it on, to map these dynamics on using a quantum classical isomorphism to the dynamics of electrons on lattices. And the specific lattices we are interested in have these so-called modes that are topologically protected. So you might have heard about these modes in other contexts. It turns out that if you set up a lattice, which has these two kinds of sites with a particular symmetry, the wave function, the ground state solutions of this Hamiltonian give you wave functions that are localized, and these wave functions are extremely robust to any kind of perturbations. And what we found in some sense is that networks like this, which occur in proofreading, they occur in adaptation, they occur in microtubules, they share they all share features that are common to or that can be mapped on to quantum mechanical models in which you think about electrons hopping on lattices. The localized mode that we are after here looks like the localized wave function in the quantum mechanical model. The transitions that we are after, or that I mentioned in the that I kept alluding to, look like the topological transitions that show up in this model. And in these kinds of models, you typically have a polarization. The electron is polarized. You have a wave, the wave function is polarized in a certain manner. Over here, energy consumption gives rise to the same kind of polarization. And so by using this quantum classical isomorphism, it turns out that if I were to look at the band structure of this of the dynamical matrix for this master equation, the band structure has a gap between the largest and the next largest eigenvalue, and this gap, it turns out, ends up protecting the steady state eigenvalue, and this is again very, very similar to what happens in the quantum mechanical systems. So this I'm obviously rushing through this right now, but I'll be happy to talk to you in detail about this. But the point the point I wanted to make was basically that it looks like there's a quantum classical isomorphism that allows us to sort of write down very, very generic principles or very, very generic design principles for how one would go about constructing networks that perform the right function. Localization maps onto localization. The localization-delocalization transition maps onto a topological transition over here, and that seems to be a useful and a productive way for us to think about these ideas. So let me end there. The first lecture was basically on how fluctuation dissipation theorems, which are the cornerstone of modern equilibrium stat mech and also nonequilibrium statistical mechanics, actually end up constraining information processing function or information processing efficiencies in biophysical systems. We then asked what happens if things go out of equilibrium, and in the lecture today morning, we took a system that is driven out of equilibrium. We didn't really care a lot about how it is driven out of equilibrium. We just had this term delta mu in there. But we saw how this delta mu can be utilized to take distributions that are equilibrium-like and transform them to a new set of distributions. So we were able to go from eta equilibrium to eta, and that's obviously applicable, or that can be used in the context of proofreading, but it can also be used in many other contexts. We also saw how writing down things like entropy production or energy consumption is useful beyond just a theoretical exercise, because this energy consumption quantity constrains fluctuations of various other quantities, and we were able to use that to come up with variational bounds in which we could use our information theoretic ideas to really predict the kind of structures that emerge in a non-equilibrium state. We could sort of use it as a design principle to control fluctuations. And in the last lecture today, we sort of saw that these non-equilibrium systems can have can have a rich class of dynamical phases that one can use to tune transport properties, that one can use to tune the kind of information processing function that one wants to achieve. So I just sort of gave you a whirlwind tour of all the things that are going on. There's a lot to learn, there's a lot to do still, and I hope these 3 lectures sort of gave you a rough overview of the state of the field and what can be done. Thank you. Yeah. Yeah. Sure. So over here, so this thing, right?
What is an epsilon? Is that something related?
Yeah, so epsilon here is, I can just write it down. Epsilon is A, let me write it down over here actually. Epsilon is A minus 0.5. So we wanted A to be close to 0.5. That's a state where the system is able to run and tumble with equal probability, right? And epsilon is how far away it is from that state. Okay? Because in some sense, as you keep perturbing the system, it's not, it sort of just goes away from the state. It doesn't exactly come back to the steady.
A is equal to 0.5, just come
No, it runs and tumbles with equal probability. So A is equal to 0 is the run, and A equal to 1 is the tumble, and A equal to 0.5 is like half and half, which is where you want it to be because it can search very efficiently there. Again, this ratio 0.5 is an arbitrary ratio. It can be any number, but I've sort of chosen 0.5 for the purposes of what we are doing. And in some sense, we want this number to be very close to 0.5 to achieve the function. Right? And so we want the system to get back to this number as you change concentrations of chemoattractant, as you change temperature, as you change a lot of things. Right? And this epsilon sort of controls that, or this epsilon captures that. And what it turns out that if you were to look and solve these biophysical control, if you were to solve the biophysical master equation for the system, you actually find that the response that you get, that's this epsilon, is intricately dependent on how much energy you pump into the system. Okay? And by pumping in more or less energy, you can actually tune how effectively you adapt to the external signal. So if this number becomes close to 0, which is what happens over here, epsilon is basically this point the system is hardly adapting. It sort of goes every which way as parameters are changed. But as this number becomes very, very large, the system is able to adapt very efficiently to external conditions. So if the external condition changes, when this if I'm dissipating a large amount of work and the external conditions change, the system responds on a short time scale, but then comes back to close to the set point on larger time scales. So that's what happens, and that's the content of this.
But these are controlled. What you can control is probably chemical gradient.
Yeah. So these are all internal responses of the system, right? So this is something that is coded into the biochemical network, and then it just turns out that if you code it in, and if you then vary the concentrations and everything else, you sort of see this kind of a behavior. Okay. So the things that you can change the concentrations, the things that you can measure are epsilons, and it seems to have this kind of a dependence. Okay. In these nonequilibrium models, and it seems to be a feature that's generic to a reasonable class of these systems. Okay, thanks.